Endotoxin shock has represented a challenge and a mystery to the physician since the time of Hippocrates. It is a threat to life for the urologic, obstetric, and surgical patient who has a gram-negative infection. The investigator uses it as a model for the study of irreversible shock in the laboratory. You have been watching the formation of an embolus in the bowel wall following the injection of 8 milligrams of difficult endotoxin per kilogram of body weight. Emboli from the bowel lodge in the liver. Because of our belief that important changes take place in the microcirculation during endotoxin shock, we have correlated a study of the microcirculation with clinical type measurements which can be made in the rabbit. Our observation that platelet microemboli occur in large numbers in all forms of shock has led us to study their occurrence in the microcirculation during endotoxin shock. Emboli forming in the skeletal muscle areas lodge in the lung. Here we see the emboli as they lodge in the lung during the acute stage of endotoxin shock. With a complete graft, we demonstrate the acute effect of endotoxin upon the platelet count. There is a drop in the first 5 minutes from 400,000 to 30,000. The platelets have aggregated and become sequestrated from the circulating blood in the form of microemboli. As emboli block the pulmonary circulation, the proximal venous and pulmonary artery pressure rises. When blood cannot flow through the lungs because of the microvascular blockage, the left heart volume diminishes and the arterial pressure drops. We have acute shock and initial cardiac arrhythmias. The bowel circulation is slow as might be expected. Clumps appear in the microcirculation as it flows more slowly.
A way of measuring the animal's metabolic energy state during the different phases of endotoxin shock in this model was by evaluating the animal's high energy compounds in both the skeletal muscle and plasma. As shown by this diagram, the high energy compound, adenosine 5-triphosphate, ATP, is universally used to propel the energy system. When involved in energy usage, it quickly is replenished by the creatine phosphate cycle by the action of the enzyme creatine kinase. Ideally, this is a delicate balance that adequately maintains itself in energy fluctuation and equilibrium. Further breakdown products are normally not expected to accumulate in a tissue. Some of these intermediary compounds, such as adenosine, inosine, and hypoxanthine, are felt able to cross the cellular membrane into the blood flow. These displaced compounds may be a tag for clinical evaluation. As you can observe in this model's first phase of shock, 5 minutes after the injection of 8 milligram per kilogram of E. coli endotoxin, a significant decline of the high energy compound ATP occurred in both the muscle and plasma. Concurrently in the muscle, the energy replenishing cycle was in full swing. Creatine phosphate was being utilized in an endeavor to help restore the ATP energy level with the accumulation of its byproduct creatine. The animal was in survival mode.
The drop of arterial pressure, elevation of venous pressure, and drop of platelet count are transient in the first phase. Recovery in the direction of normal occurs promptly. Within 20 minutes, all blood flow seems to be returning to normal. Some vessel dilatation does occur, which is typical of endotoxin shock and possibly represents a response to a persistent low-grade elevation of venous pressure. The apparent normal continues for 6 to 8 hours.
During this recovery phase, while the microcirculation improves and the platelets deaggregate, the tissue and plasma levels of ATP gradually increased towards normal. The muscle-creating phosphate level doesn't recover as promptly as ATP because it's being utilized to restore the rising ATP level, although eventually it too takes on the same course, increasing energy storage with the simultaneous drop of its predecessor, creatine.
After 6 to 8 hours, the platelet count again begins to fall. The venous pressure rises and the arterial pressure drops. A relentless, irreversible progression of events follows. In the general microcirculation again, platelets begin to aggregate. They break loose as emboli. Many will lodge in the lung. In the portal system, these emboli forming in the venules of the bowel wall lodge in the liver. The portal venous pressure rises, capillary walls break, and there is a hemorrhage into the bowel wall and mesentery. Stasis occurs and vessels thrombose. Emboli are observed to be formed in the upper limb of this venous tributary. Periodically, they flow into the vein which courses across the lower portion of this view. Higher magnification of the source from which the emboli were emitting. The platelets precipitate, they form large aggregates. These aggregates break loose and flow out through the circulation. In this terminal phase, 8 hours after the initial injection of endotoxin, we will spend a moment watching the microemboli as they lodge in the pulmonary microcirculation and block blood flow. They measure 20 to 80 microns in diameter. Circulation is blocked. If the embolus passes into one limb of a pulmonary arteriole, a shunt is formed. Blood flow is reestablished in the no longer blocked limb. This is a major mode of recovery in the first primary phase of endotoxin shock. In the terminal phase, more arterioles are blocked, and therefore the sum of the blockage is more difficult to recover from, and we have a terminal phase. In this scene, we see an embolus in an arteriole which passes from the upper corner of the film inferiorly and to the right. The embolus is lodged at this bifurcation, and gradually it will be forced inferiorly. A shunt will be formed across the upper end of the film from left to right. In this next graph, we have recorded sequential platelet counts in rabbits given 1 milligram endotoxin per kilogram body weight as dashes, and those given 8 milligrams of endotoxin per kilogram as dots. 8 milligrams is a lethal dose, and in the secondary phase of shock, the platelet count dropped below 100,000. If 1 milligrams was given, rabbits survived and the platelet count remained above 100,000. In a graph representing many experiences, we see a repeated elevation of venous pressure and simultaneous drop of arterial pressure in the terminal phase of shock. Where the bright lines cross, in each case, represents a terminal event. In the average electrocardiogram following endotoxin, one can see in B how there is an alteration of pattern with a depression of the ST wave in the initial phase of endotoxin shock. Apparent recovery occurs in C and lasts for 6 to 8 hours. In D, the ST deformity reappears, and this is at the end of the 6 to 8 hour period. In E, there is a cardiac block and ultimate cardiac standstill.
One main player in this whole ordeal is the high energy molecule ATP. Energy is a source that supports life. Whenever the rabbit's vital signs begin to deteriorate, the chemical energy level begins to steadily diminish in both the tissue and plasma. As the microcirculation becomes compromised, the energy level drops and metabolic failure manifests itself. Again, to reiterate the first phase of shock, during the first platelet aggregation, the creatine level increased by the removal of a phosphate bond from the creatine phosphate molecule, trying to restore the vital ATP energy usage. In partial recovery, with the platelets deaggregating and the restoration of the microcirculation, the energy storehouse creatine phosphate was restored. As the secondary phase of shock set in, with a secondary drop of the platelet count and the increasing vascular blockage, both the muscle creatine and creatine phosphate levels were irreversibly lost. The energy storage safeguard was being eliminated. The rabbits had no breathing support, and they hyperventilated in an attempt to compensate for the induced metabolic acidosis. As a result, arterial oxygen tension was raised during the early periods of shock and began to decrease after 7 hours. The carbon dioxide tension fell steadily from the very beginning. Carbon dioxide tissue generation represents cellular energy production. A decrease of tissue CO2 output is a sign of tissue metabolic insufficiency. When tissue oxygen is in short supply, portions of pyruvic acid may not enter the Krebs cycle, but rather is changed into lactic acid and then diffuses into the bloodstream. As shown in this diagram, there was a continuous rise of lactic acid levels in both the tissue and blood from the very beginning of shock. After the injection of E. coli endotoxin, a decrease of blood bicarbonate, an increase of blood lactate, and a rising base deficit are all related to oxygen transport imbalance at the cellular level. Even though there appears to be a vascular recovery portrayed by the blood flow observations and high energy tissue measurements, previously observed, there remains a continuous decline in tissue metabolism portrayed by the blood gas analysis and rise in the lactic acid levels in both the tissue and blood. Thus, partial recovery may only be a term designated for a hopeful outcome. Despite the energy imbalance, the blood pH was successfully maintained until 7 hours after the injected endotoxin. At this time, the blood buffering system began to fail, which led to a gradual fall in pH during this terminal stage. Returning to the nucleotide study of the muscle and plasma, let's again look at the byproduct of ATP. In a blood clinic, adenosine 5-diphosphate, ADP, is used to aggregate platelets in an optical aggregometer for testing their ability to adhere to each other. In the study, as the muscle ADP level decreased, the plasma ADP level increased. At the vascular endothelium border of this tissue-blood exchange, high levels of ADP may have existed, instigating platelet adhesion on its walls. This could have implications in the secondary platelet drop. After the injection of endotoxin, early intermediary products of muscle ATP breakdown occurred. The nucleotide adenosine 5-monophosphate AMP and inosine 5-monophosphate IMP elevated as early as 1 hour. This increase extended over a 7-hour period of time. The nucleoside adenosine did not significantly increase in the muscle. The plasma levels of all three of these compounds never significantly varied during the entire experiment. After 7 hours, the late decline of muscle AMP and IMP could have been responsible for some of the measured gains of inosine and hypoxanthine in both the muscle and tissue. Hypoxanthine can be a dangerous molecule when accumulating in tissue or plasma. Normally under oxygen-friendly aerobic conditions, it can be degraded by the enzyme xanthine dehydrogenase into NADH plus the hydrogen ion, which feeds back into the energy-forming Krebs electron transport chain and urate, which is eventually eliminated by the body. Under low oxygen tension or ischemia, hypoxanthine is broken down by the action of the enzyme xanthine oxidase, producing destructive oxygen radicals which can destroy vital tissue structures. As you can observe by this diagram, after a lethal injection of E. coli endotoxin, an early 5-minute rise of hypoxanthine occurred in both the muscle and plasma. The nucleoside inosine, which can be broken down into the purine hypoxanthine, had an early rise in muscle but not in the plasma. In late shock, inosine was substantially elevated in both the muscle and plasma. In partial recovery, when adequate blood flow resumed with platelet deaggregation taking place, both inosine and hypoxanthine levels diminished towards normal. In late shock, there was a surge of both inosine and hypoxanthine in both the tissue and plasma. As previously indicated, hypoxanthine can be a dangerous molecule under ischemic conditions with potential radical formation. Tissue feeds the venous circulation, with a draw across the cellular membrane. In contrast, the arterial flow delivers contents back into the cells. A simultaneous comparison of the plasma hypoxanthine concentration in 2 rabbits at various times before and after a lethal injection of endotoxin demonstrated that hypoxanthine is normally degraded in the tissue, exposing it to the action of xanthine dehydrogenase, or under ischemic distress, xanthine oxidase. This gradient was consistently maintained during all the time measurements. In an overview on the muscle tissue loss of energy producing molecules, the adenine nucleotide pool, ATP and ADP, declined 46% in the terminal phase of shock compared to normal. The total creatine pool, creatine and creatine phosphate, lost 48%, and the combined total adenine purine pool, ATP, ADP, AMP, IMP, adenosine, inosine, and hypoxanthine dropped 45%. Even if restitution was possible at this time, such large losses of energy-producing metabolites would be difficult to restore. With a lack of available oxygen in the cells due to obstructive interference, the cells need to replenish energy by anaerobic means, which resulted in accumulation of lactate. The terminal muscle level of lactate increased around 2.7 times normal at the late phase of shock. Creatine phosphate is a highly charged molecule and does not excessively cross the cellular membrane. It's needed to replenish the ATP level and therefore the darling of the cellular cocktail. As shown in this diagram, after the injection of a lethal dosage of E. coli endotoxin, the normal plasma creatine phosphate consistency was maintained in the first phase of shock and only began to elevate after 3 hours and was significantly increased at 7 hours. Within the terminal phase, plasma creatine phosphate was elevated approximately 4 times normal. This surely suggests that the integrity of the cellular membrane had been compromised and damage inflicted. Monitoring plasma creatine phosphate levels could be a tag for cellular membrane damage. We watched platelets aggregate from the vascular walls and observed them blocking the arterial bifurcations, blaming them for tissue ischemia and eventual tissue failure. They appeared to be an important visual culprit in the demise of the animals. One thing we didn't discuss was the clotting cascade and the formation of fibrin mesh. At 5 minutes after the injection of a lethal dose of E. coli endotoxin, with the dramatic drop of the platelet count, there was not a decline of the plasma fibrinogen level or significant increase of the factor VIII clotting time or any detection of fibrin split products. The clotting chain at this time appeared not activated. But still, the arterial pressure lowered, the central venous pressure rose, and tissue ischemia occurred as demonstrated by the metabolic studies. Even 1 hour later, during our partial recovery phase, the fibrinogen plasma level was uncompromised, factor VIII levels remained in range, and fibrin split products still were not detected. Later, at 7 to 10 hours, during a second platelet drop, there emerges a different pattern. The clotting system had become activated, possibly directly by platelet thromboplastin or by tissue factor liberated by tissue injury. During this time, the fibrinogen plasma level dropped 55%. Factor 8 time increased from 35.9 to 52.5 seconds, it being utilized in the clotting process, and fibrin split products were detected. The breakdown of fibrin by the blood plasmin was likely a vital attempt to dislodge vascular obstruction. Near death, there was almost a doubling of the factor 8 time, a 62% decline in the fibrinogen plasma level, and a further detection of fibrin split products. These results confirm that disseminated intravascular coagulation plays an important role in endotoxin shock pathology.
In summary, we have demonstrated that platelet aggregates form in an early and terminal phase of experimental single-dose endotoxin shock. Microemboli lodge in the lung in both phases, block the flow of blood, produce an elevation of venous pressure and a drop of arterial pressure. In a mechanical way, this can lead to microvascular stasis, proximal thrombus formation, disseminated intravascular coagulation, pulmonary congestion, pulmonary shunts, oxygen and carbon dioxide exchange problems, right heart failure, arterial hypotension, and cardiac arrest. This view is a terminal view of the circulation in the bowel wall in endotoxin shock.